Perfect.
Thank you very much, Sam, for agreeing to give a Build a Cell seminar, and you can get started.
All right. Hi, everybody. I'm Sam Schaffter. I'm a chemical engineer in the Cellular Engineering Group at the National Institute of Standards and Technology. And then this talk today will be primarily focused on co-transcriptionally encoded RNA strand displacement circuits that we've been developing for applications in engineering biology. And maybe before I get into the technical portion of the talk, I wanted to give a little bit of background on the Cellular Engineering Group as a whole, and sort of my background and how we're approaching problems in engineering biology. So the Cellular Engineering Group is a relatively new group at NIST. I think it was started around 2015 or 2016. And one of the big things that the group has sort of been focusing on is trying to build living measurement systems. So engineering cells that can then sense and respond very precisely to different environmental inputs. And so there's sort of three different focus areas that the group has sort of been focusing on towards these efforts of engineering living measurement systems. So the first one is developing sensors to sense different things in the environment. And so a lot of that work is headed up by David Ross in the group. And he does a lot of protein engineering projects where they're designing allosteric transcription factors to sense small molecules inside bacteria, as well as some work on GPCRs to sense extracellular components using yeast. And then another big effort in the group is designing different chassis for engineering purposes. And some of that work is on using cell lysates from different organisms to prototype how parts are going to work. And so that's a lot of that is headed up by Jane Romantsova in the group. And then there's a lot of ongoing work engineering minimal cells that is headed up by Elizabeth Strahalski. And so then that leads to the last part of what we've been working on, which is a sort of molecular computation piece. And that's sort of where I come in. I joined the group as a postdoc back in August of 2020 and wanted to head up these efforts of trying to engineer molecular computation to sense different combinations of inputs in the environment and sort of make sense of that information in some programmed way to ultimately tell the cell what to do. So before I joined NIST, I was a graduate student at Johns Hopkins in Rebecca Schulman's group, and I worked primarily on DNA computing and DNA nanotechnology projects. And so when I was transitioning into my postdoc, I wanted to take a lot of these principles of DNA computing and DNA nanotech and try to apply them towards a more cellular engineering type of application. So that's a little background on me and our group as a whole. And now I'll sort of jump into this idea of building molecular computation for engineering biology. And really, molecular computation is sort of a hallmark of living systems. Cells are always sensing, processing, and then responding to changing stimuli inside in their environment. And so there's a growing effort to try to be able to do this from the bottom up using molecular circuits. So a big effort to develop programmable, predictable, and very modular molecular components that can be used to process information either in a synthetic cell or engineered live cell application. And so there's obviously lots of different ways to go about doing this in these different applications. And I won't belabor too much of that and talk about all the different possible paradigms for doing molecular computation. I'll just sort of cut to the chase of how I approach this problem, which is using RNA-based molecular circuits. And specifically, I work with circuits that operate via a principle known as toehold-mediated strand displacement reactions. And so as I mentioned, my background is more in DNA computing. So traditionally, these strand displacement components are made of DNA, but the same general rules apply for RNA. And there's basically two kinds of components you have in these systems, these single-stranded nucleic acid inputs, and then these partially double-stranded nucleic acid gates. And each gate can be designed to have this little overhang known as a toehold. And then through sequence complementarity, this toehold can recruit this input to bind and then initiate a process of branch migration that ultimately displaces this output strand from the gate complex. And the idea is now this output strand has a new unsequestered toehold, and so it can serve as an input to another strand displacement reaction downstream. And what's nice about this paradigm is you can sort of abstract quite well away from the sequence level, and you can sort of say, hey, I'm going to program any arbitrary set of reactions like A plus B goes to C, and you can more or less do this quite reliably by just designing complementary sequences. So this serves as a sort of nice molecular programming language that lets you sort of abstract away from a little bit of the molecular details when designing these circuits. And beyond this level of abstraction, you also have very nice control over the kinetics of these reactions. So by just changing the sequence or length of that toehold that initiates strand displacement, you can control the kinetics of the reactions over roughly 7 orders of magnitude. So this gives you a lot of control on designing different types of reactions that can threshold out inputs and things like that to do digital logic and things of that nature. So the field of DNA computing is taking advantage of these nice properties of toehold mediated strand displacement. And it's used this to build very scalable and programmable circuits. So people have built circuits composed of hundreds of different strand displacement components that can do programmed things like compute the square roots of numbers that are encoded in binary, or recognize patterns of up to 100 different DNA inputs that are quite sophisticated. So this really shows sort of how this is a very powerful bottom-up way to construct molecular circuits for information processing in the biological realm. One sort of limitation for using this technology, especially in closed environments like synthetic cells and living cells, is it's difficult to genetically encode these components. And really this sort of stems from the way that everything is designed, where if you have multiple gates that are designed to react in a cascade, they have a lot of sequence complementarity with one another. And so you basically have to produce each gate in its own separate test tubes and anneal those separately so that they'll be kinetically trapped before you can mix all the components together to make the final circuit. So it is possible to build circuits like this and then either encapsulate or transfect those into these closed systems like a synthetic cell or a living cell. And there's been a lot of work to do this kind of thing. But one limitation with this approach is that the circuits are still sort of a single use computation. Once you've done the calculation, you've used up all the circuit components, and you either have to re-encapsulate or transfect those back in if you wanted to do another calculation. Another problem is that they sort of have a limited lifetime. If your input doesn't come around quickly, after you've done this transfection, the components are susceptible to degradation, and eventually, you'll basically degrade everything if you wait around long enough. So we've been trying to come up with some ways to sort of alleviate these kinds of limitations by asking the question, can we find a way to sort of genetically encode RNA-based versions of these circuits so that we could operate them continuously in these closed environments? So the idea is if you could sort of encode these genetically, now you can continuously produce these parts in these different environments in situ and sort of combat the degradation issue and continuously replenish parts so you can do multiple use computations. I think there's a lot of other advantages to being able to genetically encode things versus just transfecting components in there, because genetic encoding allows you to sort of have this inducible control over when and where you express different circuit elements in these environments, and allows you to sort of integrate with a lot of other genetic circuit elements, which I'll touch on a little bit at the end of the talk. So thinking here about how to genetically encode these components, there's really one major question that we need to address is, how do we actually produce these components together in the same place without having cross-reaction between partially complementary components? And so for that, we've developed this technology that we call cotranscriptionally encoded RNA strand displacement circuits, which allows you to sort of do this genetic encoding of RNA versions of strand displacement components. So the idea is now we're going to have DNA templates as sort of the input into the system, and those can be transcribed to produce either an RNA input or a RNA gate. And sort of the trick to getting this to work so that you can produce multiple gates with sequence complementarity together in the same reaction is we design the gates to initially be produced as a single RNA that can fold into a hairpin, and then it contains this self-cleaving ribozyme motif. So after this RNA hairpin is folded, it can undergo this cleavage reaction and make the final double-stranded RNA gate that's suitable for strand displacement. And the idea is this sort of folding is going to happen during transcription, so very quickly, and this should allow these gates to be produced and not cross-react with one another, even if they're designed to react in a cascade. So here's an animation of how this process is supposed to work, where we're transcribing this gate. It should be folding while it's being transcribed. After it's folded, this ribozyme cleavage step can occur, opening up an input toehold on the gate, and this can now recruit the input to bind and initiate this process of strand displacement. That ultimately kicks off this output that can now be programmed to interact in other strand displacement reactions downstream. And as I mentioned, so the key here is now we can produce multiple gates that are designed to react in cascades, but we can do this together in the same reaction because these gates should sort of fold independently during transcription and not cross-react unless you have the correct input present to initiate the strand displacement cascade. So that's the general idea of what these cotranscriptionally encoded RNA strand displacement circuits are supposed to look like and how they're supposed to operate. So we next needed to go about actually building a system that hopefully has these desired properties. So we went about designing a few different sequences and testing out a couple of different designs, and finally settled on one that seemed to work pretty well. So here I'm showing gel electrophoresis results, both native and denaturing, to show that these gates seem to fold the way that we designed them to and have very high cleavage efficiency. So over 90% of the transcripts that we produce end up cleaving the way we designed them. And then we were able to get some estimates of this cleavage rate constant that are sort of consistent with this model of the reaction, where essentially ribozyme cleavage is the rate limiting step in the gate maturation process. So we next wanted to look at characterizing the kinetics of this reaction. And for this, we've been doing everything in a simple in vitro transcriptional setting. So all the results I'm talking about today are still in bulk reactions in a simple in vitro transcription scheme. So we just have these linear DNA templates, some magnesium buffer, and then NTPs and T7 RNA polymerase in these reactions. And so the templates should be transcribed by T7 to make these RNA inputs and RNA gates. If those two components are complementary, they should undergo this process of strand displacement, kick off this output sequence, and then we can measure the production of output as a function of time with this nice fluorescent output using this DNA reporter. So it's basically 2 strands of DNA modified with a fluorophore quencher. If the output is released, it can initiate strand displacement to sort of kick off this fluorescent strand from the reporter complex.
We lost your screen sharing for a second there.
I don't know what happened there. Let me try again. All right. Is it? It's back?
Yep, it's up. It's back. Thank you.
All right. So I was just talking about the DNA reporter. So that's what we're looking at in these results that I'm showing here. This is reactive reporter percent as a function of time. And so you can control the kinetics of this reaction by varying the ratio of input to gate template in this system. And importantly, if we have this control line here where we've put in a scrambled RNA input, so it's not complementary to this gate, and we see very little signal coming out from that control there. So this shows our ability to sort of characterize the kinetics of this. The next big question is, how well can we actually predict these kinetic trajectories using a simple model of mass action kinetics? And this is a key sort of piece of being able to use this technology, because a big factor in DNA strand displacement circuits is you have very predictive control over how those are going to behave. And so that allows you to just sort of do this abstraction away from a lot of the molecular details to program much higher order function. And so we wanted to be able to have a very similar predictive control over this system if we want it to be as useful as the DNA-based versions have proven in the past. So basically, you can enumerate all these reactions out that we've designed to be present in the system. You can set those up to be a set of ordinary differential equations for mass action kinetics, and then integrate these to look at how these different species evolve as a function of time. So when we do this, you can get fairly good agreement with our kinetic trajectories at these higher input to gate ratios. But one thing that we're really missing is this leak term in the model, as our model doesn't have any terms that should suggest there should be any output being produced if you don't have the correct input present. And so, there's no term to capture that, what we see in the experiments in this current model. So we next wanted to say, all right, what are some plausible mechanisms by which this leak could be occurring? And can we include those terms into our model to get a better predictive capability? And there's basically 3 different leak mechanisms that we were looking at here. So one is a common way of having leak in DNA-based systems is sort of a reaction between the gate and the reporter that doesn't really use much of a toehold region. It's sort of a zero base pair reaction. And this often happens in DNA-based systems. And so we thought maybe this is going on in our system as well. Another reaction that could be leading to leak is perhaps this transcript can react with a reporter before it's had a chance to fold. So this is sort of, what I was initially thinking might be going on, that perhaps our assumption that co-transcriptional folding is going to happen so quickly that there won't be any reaction is perhaps wrong. And then the last model that we looked at is one where perhaps some fraction of these gates that we're producing are either truncated or misfolded in some way, so that they basically have the output toehold already exposed. and then that can react directly downstream. So in other words, this is sort of like some fraction of the gates are basically just produced as functional outputs, and that's leaking in the system. So we could take our original model and then independently put in these 3 different reaction terms to see how that would change our predictions. And so for model 1, we don't get very good agreement here. You basically need to accumulate a lot of this gate transcript before you start to see the leak. That's sort of inconsistent with what we see in our experiments. But for the leak model 2 and 3, both of these seem to be feasible and can recapitulate our results quite well. But obviously they're much different mechanisms. So we next wanted to design an experiment to see which of these 2 models might be more consistent with what's actually going on in the system. So for that, we designed this sort of simple experiment where we wanted to transcribe the gate by itself for different amounts of time. So half an hour, 1 hour, and 2 hours. So basically we produce increasing amounts of RNA in these different test tubes. Then we can denature the polymerase and then add in our DNA reporter reaction. So it's sort of a very crude extraction of RNA followed by addition of this reporter to see how those crude extracts of RNA react with the reporter. And what's nice is then if you model this simple experiment, you get 3 different unique output profiles that we might expect depending on which of these models, leak models, is actually occurring. So for leak model 1, you sort of expect a 0 signal at the beginning and then a steady increase as a function of time with an increasing slope the longer you've allowed transcription to go before you've added the reporter in. In leak model 2, presumably at the end of the experiment, we would have had all these RNAs folded, and so you wouldn't really expect much of a leak signal in this case because you're not actively producing the gate anymore. You have already folded gate. And then in the third model, you have these truncated amounts of components that can sort of react quickly downstream like an output. So you sort of expect this rapid increase in signal, and then the amplitude of that is higher the longer you've allowed transcription to go because you have more, you've accumulated more of these truncated products. So this is sort of the idea of what this experiment should look like. And then when we run it, it looks the closest to leak model 3, where we sort of get these discrete increases that increase in magnitude the more RNA we have. So that seemed to be the most consistent leak model with what we've got going on. And I will say there's obviously always caveats to this. I think the leak is maybe a little bit more complicated than we have it described here, but putting this into the model gives us pretty good predictive capabilities, even though it's probably not entirely complete on the exact mechanism of what's going on. And I think one interesting takeaway from this is in this leak model, to recapitulate our experiments, we only really have to have 3% of the gate transcripts being incorrect to recapitulate this. So in some sense, a large fraction of our RNA does appear to be correct. We sort of just accumulate this at a slow rate in the system, which ultimately leads to a relatively high leak at the end of the experiment. Because again, there isn't any degradation in these things, so any truncated or misfolded products that we make just continue to accumulate the longer the experiment goes. All right, so that's sort of the wrap up of the modeling section. And now, I've spent the majority of this talk only talking about this single sequence. So the big question is, can we make more sequences, and how well do they, conform to what we've learned from this single sequence? So that was the next big step. Can we scale up the number of sequences so that we can start to build more complex circuits? So what we did is design gates with 16 different input sequences. So that's what this X domain is. It's one of 16 different sequences. And then we wanted to assay these sequences performance. And from the previous strand displacement literature, there is some expected variation in strand displacement rates as you change these sequences. So we use our simulations to sort of come up with the shaded region of sort of where we might expect the distribution of strand displacement rates to fall. So the idea is anything that's sort of outside of this shaded region is definitely behaving in a way that's sort of worse than we would expect from just natural variations in this sequence. So we designed all these 16 sequences and then tested out these gates performance. And so we find many of these gates work, but maybe unsurprisingly, not every single one of them works the way that we wanted them to. So a number of different gates had much slower strand displacement kinetics than we would have expected from our simulations. And we also find a number of gates actually have very poor ribozyme cleavage efficiency, just depending on which sequence is adjacent to the ribozyme. So we wanted to spend some time trying to understand these mechanisms a little bit more to see what's going on here and see if there's a way that we could maybe improve our design moving forward to sort of avoid some of these these pitfalls that we were seeing. So I spent, maybe 3 months or so looking into these different sequences to try to understand what was going on. And one big failure mechanism that we found is using repeated toll holds sort of increases the chances that a gate of a given sequence combination will misfold. So repeated toll holds is something that's commonly done in DNA strand displacement circuits where the input toll hold and the output toll hold are both the same sequence, shown here as U prime. And this is nice because it makes the circuit very modular. Everything's sort of initiated with the same sequence, and it allows for very similar kinetics between everything because they're all initiated by the same toll hold sequence. Now, when you do this with DNA, you're sort of going to thermally anneal all of those components together. So they typically adopt the designed secondary structure that you want. Problem with our system is we're actually transcribing this thing and it needs to fold while it's being transcribed. So if you have these repeated sequences, you can basically introduce other alternative folding pathways that might disrupt ribozyme cleavage or maybe the strand displacement kinetics, for example. So we think one general rule is repeating toll holds is not a good idea in this RNA-based co-transcriptional regime. Another big failure mechanism we found is, as I mentioned, many sequences actually had poor ribozyme cleavage. If you look a little bit closer at the sequence of the ribozyme, it has some sequence motifs that are sort of inconsistent with some of the other sequence constraints we typically use in this system. And so, we wanted to identify if there's other ribozyme sequences we might be able to use that would be less prone to misfolding in our context. So we updated a number of sequences, found some compatible toll holds that could be used with similar rate constants, and then designed a suite of different RNA sequences that are either taken from other organisms but have similar folds to the one we used, or were a few point mutations on the ones that we had tested in the past. And then, in addition to updating these sequences, we sort of came up with a nice flow chart of, recommended design approaches of try these sort of design tricks first, and then depending on the failure mode, this is how you should update the design to do a better job moving forward. So using these new sequences and design approaches, we were then able to recover the performance of all 16 of these sequences that we initially screened to get them to fall within the behavior that we expected. And this is, again, one sort of sequence context where basically the only thing we're changing is X and everything around it is the same. So we wanted to see how well this actually works for a broader range of sequence contexts. And so we designed 35 of these 2-layer cascades, where basically all the gates in the second layer of the cascade have been designed to have a number of different input sequences, and then they have different output sequences. So this is effectively a wide range of 35 different sequence contexts where we've been testing these design approaches. And so we get very good performance here, falling pretty well within the region of what we expected from our simulations. So this gives us some confidence that these are fairly reliable design approaches for putting this toolkit together to build individual circuit elements. All right, so now, we've developed this CTRSD circuit paradigm. We have a large set of sequences and a set of design approaches to sort of tell you how to put these sequences together. So can we actually start integrating these things together to build more programmable circuit elements? So we started with programming just simple logic elements. So here's some examples of 2-input and 4-input OR gates. We've also designed a number of 2-input AND gates. We're going to get pretty good ability to control these and predict their behavior with our simple model. And then once you have these elements, you can start to integrate them together to get more complex multi-input logic by cascading these elements together into more complex circuits. And then we've tested 2, 3, and 4-layer cascades to sort of show that you can program these at varying depths. And so the more layers you have, maybe the more capabilities you have to program higher order functions moving forward. So the last thing we've been working on recently is trying to build slightly more complicated RNA-based circuits, ones that might be able to recognize and classify different molecular pictures, for example. So this is an example of a 9-input molecular perceptron. This circuit had been developed previously with DNA computing, and we just wanted to map that same circuit architecture over to the CTRSD paradigm to see if we could get the same kind of programmable capabilities there. So the idea behind this circuit is we basically have 9 inputs, and you can map each one of these inputs to a pixel in this 3x3 grid, and then you can define patterns that you want to be able to distinguish with the circuit. So for example, we're going to use X and Y, where these filled in squares are the presence of these different input patterns. And what's nice about the circuit is you can sort of program it so that it will be able to recognize which pattern it's closer to, even if you don't have a combination of inputs that's an exact match. So you might be missing a few pixels or have a few added pixels that can still tell you if you're closer to X or Y. So it's effectively a linear classifier for these 9 input patterns. So we're able to build this circuit using CTRSD, again, just mapping what had been done with DNA computing directly into our transcriptional coding encoding paradigm and get pretty good predictability for this system. So this integrates 9 different inputs and 15 gates together and can do this classification calculation for us. So that's pretty promising results that we can just take existing circuitry from DNA computing and map it to our technology. All right, so to summarize this portion of the talk, we've developed these CTRSD-based circuits. We have a nice toolkit of sequences and design approaches that sort of tell you how to put those things together so that each part will function with a high chance of success. And then that allows you to build these nice predictable and programmable circuits with any of these different functionalities. And as I mentioned, I think a key advantage to this approach is we don't have to, reinvent the wheel. There's a lot of great work going on in DNA computing. People are developing different types of circuits all the time. And the idea is we can sort of map those directly to the CTRSD paradigm and sort of piggyback off a lot of the great work that's been done there, but in a way that allows us to sort of genetically encode these kinds of components. And as I mentioned throughout, we use this simulator a lot to understand how these circuits should behave and to allow us to build more complex functionality. So I had an intern surf student last summer, Terrence Murphy, who helped me put together a more user-friendly version of this simulator. So we have a nice website that's linked here that goes through, how to use all this, all the different reactions that are present, and how to use this to sort of abstractly simulate the kinetics of any sort of arbitrary CTRSD circuit configuration. And it also has a sequence compiling component, so you can then compile the DNA sequences you would need to order to test those out. So we're hoping this will help make this technology easier for other users to adopt moving forward. All right, so I have a little bit of time left, and I wanted to talk briefly at the end here about some future directions that we're exploring and some other ongoing work related to this technology. And so, as I mentioned, one thing we've really been focused on now is testing this out in the simplest possible system, these simple in vitro transcription reactions. So the only thing we have going on is just production of RNA. We wanted to expand the capabilities by maybe adding different RNases into the system so we could programmably degrade different components. And so we can have this sort of production degradation process where maybe we could build a system that's sort of maintaining an out of equilibrium steady state that could be perturbed as you change different inputs in the system. So this is ongoing work in collaboration with Rebecca Schulman's group at JHU. And then, trying to go into more complex systems where transcription and translation can occur, like the simple pure express system, or even in-house made cell lysate systems. Again, this would be with Jane Romantsova at NIST. She's sort of the resident expert on cell-free expression there. And we can use those types of systems to then understand how this might work when we go into cells, thinking about microbes and yeast, and maybe in the future, even more complex eukaryotic cells. And I think along the way, there's lots of interesting use cases where encapsulation or localization or the use of synthetic cells might bring about new properties and give us a lot of interest, or give us a lot of capabilities of prototyping some of these more complex systems. So you might imagine in these cases where you can have production and degradation of RNAs, that you could build local gradients with these synthetic cells, build pretty complex out of equilibrium type systems. We could also use these as sort of prototype how encapsulation in a much more controlled environment might influence the different reactions that are occurring in our system. So this should inform how these circuits might change when we try to take them to cells. Another thing that we want to do is sort of expand what inputs and outputs these circuits can take. So right now, it's sort of limited to RNA inputs, RNA computation, and then production of, in a sense, some sort of quasi-useless short RNA output. So we would like to expand this to be able to do more types of inputs and outputs. So on the input side, trying to couple this to small molecules or proteins, and then doing the computation based in the RNA space by sort of just modularly converting between these other types of inputs to RNA. And this is where I think having the ability to genetically encode everything, give you some functionality, because you could imagine for the small molecules, basically just having allosteric transcription factors that will de-repress the production of the RNA inputs into our system. And so this sort of allows you to easily convert between small molecule into the RNA space, where you can do this very programmable computation on those patterns of inputs. On the output side, you might want to be able to use these outputs to actually regulate gene expression. So we're looking at coupling this with a number of existing RNA-based technologies that allow you to regulate gene expression either at the transcription or translational level, so that you can get functional protein outputs after you've done the calculation in the RNA space. So hopefully I've convinced you that these CTRSD circuits are a fairly versatile platform that could be used across a bunch of different environments to hopefully give you an ability to target lots of different applications where you might have been somewhat limited with using just the DNA paradigm. Next. Now, I have one more tiny vignette. Since I am from NIST, you got to have some brief discussion of measurements in any one of your talks. And so, I've sort of laid out this idea of increasing the complexity in the environments to sort of systematically understand how that changes everything, ultimately allowing us to get to operation in cells. And I think as you go into these more complex environments, you're going to need measurements that sort of will work across all of these different spaces. And one thing that I'm sort of interested in is I think a lot of times people focus on the circuit output. We just need to measure if it's doing what we think it's going to do in these different settings, and then that's sort of good enough. But I think another key thing, especially in these RNA-based circuits, is having ways of looking at what the RNA structure is, having measurements for RNA structure so that you confirm not only does it work the way you think it does, but it's actually doing it via the mechanism that you designed. I think that's sort of critical for being able to really engineer these components moving forward. And the reason I sort of am talking about this is because I already have some data in hand that sort of shows the importance of where looking at just circuit function can sort of be misleading. So here's 4 different RNA strand displacement gates that I designed, and these are the kinetic trajectories from our simple DNA reporter assay. So you can see they all have very similar performance. They get very quick kinetics when you have the correct input shown here in the filled in brown lines, and then they have pretty low leak within the region that we normally expect when you have this scrambled input shown by the semi-transparent lines. But 2 of these gates cleave greater than 90%, and 2 of them cleave very poorly, less than 25%. And if you just look at the function here, you'd say, hey, all 4 of these are functionally equivalent. I can go ahead and use them. We actually know 2 of them aren't folded in the right way that they've been designed to fold into, because the ribosome isn't actually cleaving. So, I have some theories about exactly what's going on here, but I don't know entirely what it is. But I think the idea is that, you want some way of measuring structure in these systems. In our case, we've been using ribosome cleavage as sort of an indirect way of telling, if something doesn't cleave, it's obviously not in the right structure. And so we know this is a failure mechanism. We've been wanting to find ways to do this measurement that can work across all these different environments. And so we have some recent work that should be wrapping up soon, where we've been using RT-qPCR-based measurements to do measurements of ribosome cleavage. And this is sort of environment agnostic. You can express these RNAs in cells or in Pure Express or whatever, extract that RNA and then do this kind of measurement on those components to get a sense of what's going on. So we've been benchmarking that using RNAs that we have orthogonal cleavage measurements for, using denaturing gel electrophoresis. And then we've recently been expressing these gates in E. coli, extracting total RNA, and then rerunning this RT-qPCR assay. And not surprisingly, we do find differences between RNAs that are transcribed in vitro versus those transcribed in cells. Basically, we get a little bit better performance, it seems like, when you do this in E. coli cells compared to the in vitro transcriptional setting. And moving forward, we'd like to do more direct measurements of what these RNA structures are, so we can learn more about what's going on when things misfold. So using things like ShapeSeq or DNS MapSeq basically allows you to do chemical modifications of unpaired bases, and so you can get a sort of more direct measurement of what the base pairing is in these systems, and hopefully use that to inform more predictive algorithms to design sequences moving forward. So that's all I have. With that, I'd like to acknowledge all the members of the Cellular Engineering Group that have helped make this work possible. As I mentioned, Terrence did a lot of the programming for our sequence simulator. Nina has done all of the RT-qPCR experiments for me, and Olga does a lot of cloning and things like that for me. And everybody else is always invaluable in terms of discussing and mapping out projects. and everything in that space. And finally, a quick shameless plug that the group is always recruiting and looking for collaborations. So if you're interested in any of this RNA-based work, definitely reach out to me. And then we have a number of other ongoing projects as well that I sort of talked about at the beginning. So if any of those interest you, you can either contact me or the project leads directly, and we can talk about that. With that, I'd be happy to take any questions about any of this work or any thoughts on potential collaborations or anything like that.
Thank you. Thank you so much, Sam. There is a lot of questions in chat. Can you see chat?
Yeah, let me.
If you can read a question out loud before answering it, that's going to be for the recording.
All right. All right, so the first question is, what is the limit of how many orthogonal strand displacement gates you have in the same tube, and is there a way to design them long enough that you can anneal them all in the same tube? I would say, theoretical limit, I'm not entirely sure. Based on the DNA computing space, they've done this where they've gotten up to 100 different components all working together. Again, though, you have to make all these components sort of separately and then mix them together. So in principle, I think we could have a similar level of orthogonality in terms of the sequences. There's some, things that become an issue, especially when you repeat a lot of toehold sequences. You can sort of get a bunch of non-specific binding the more of those you have. But I would say, if it can be done with DNA, we should be able to do it in principle in terms of orthogonality. I don't know how easy it would be to genetically encode hundreds of different RNAs and operate those in cells, but I don't know that the crosstalk there is the exact problem. The next question is, do you have a problem with RNA stability, especially for short RNAs? Do you need to be able to do any tricks of inhibiting nucleases? Yeah, so as I mentioned, most of our work now is still just in a simple in vitro transcription setting, so we don't have any RNases in there, unless I've accidentally, included them somehow with my handling. One thing that is somewhat tricky is we have, the outputs are single-stranded 3' ends, and so I think those are less stable in cells. We have some evidence of that. But there are tricks that we can play to basically like add 3' hairpins and things to different components. So that would be one way I would imagine to modulate RNA stability in different settings, is change what the 5', 3' hairpins are on the RNAs. In terms of doing things in cell lysates, we would probably add RNase inhibitors to try to remove that variable in those settings. But in some sense, we do want degradation, especially in cells, because you sort of want the circuit to continuously turn over. It's just a matter of understanding, what the relative stability of all the different components are. Next question is, what temperature do you run the experiments in the simulation? Can you use the temperature to modulate it? So everything I've done so far has been at 37 degrees C. The simulations don't really have a parameter that you could change for that. Basically, all the other rate constants would change if you change the temperature. So like if you went down, for example, to 25, I think transcription would get a lot slower. And then, that would be an issue. The only thing I know for temperature is some of these gates that misfold and don't cleave, if you just heat them up a little bit to like 42 degrees C, they then do cleave. So it seems like whatever they've misfolded into is maybe some sort of metastable kinetic trap. And if you prod them out of it, you can get better performance. I have never tried to then transcribe anything at 42 degrees and see how that works. That would be an interesting, study that just haven't done. Next question, could you maybe increase RNA degradation to keep the leak low and then make the system more dynamically responsible? Yeah, so this is, I think I mentioned this at the end. You probably put this question before I got to it, but we are actively incorporating RNases into the system to try to do this very thing, where we would sort of degrade the leak products RNAs and then maintain some sort of steady state. And the idea would be, if you have a steady state, you could perturb it with some set of inputs. If you take those inputs away, it should reset to the steady state, and now it'd be ready for another set of computations. So another question, which is a good one, is NIST going to develop anything close to strand displacement standards, anything like industry standard for implementing it, for example, to control bioreactors? Is that even worth doing? Or each applications would be too different? Well, I think this is a great question. And maybe if we could get enough people together that were, stakeholders in this idea, we could try to figure out if it would be worth doing in some standardized way versus looking at different applications. I know there's a lot of sort of domain specific knowledge here that might be good to get, like maybe the beginning thing would just be some sort of documentary standard on how to run strand displacement reactions and maybe having tips and tricks of the trade. So I think that's a great idea. I'm going to write that down as something to discuss with the rest of the group. Next question for the cell-free system work. As you work on precise measurements, how do you account for batch-to-batch variability? I think this is an open question. One thing that Jane has been working on is developing a sort of reference plasmid so you can kind of calibrate what the activity of your cell lysate is. So in some sense, we would use that to know what those differences from batch-to-batch variability are, and maybe in terms of transcription rate and translation rate. But yeah, in terms of being able to reduce that variability, I think that's sort of an open question. It might just be having to use that to sort of calibrate what the expected performance is. So one thing I do is I have a lot of, well, not a lot, but there is variability from batch to batch in T7 RNA polymerase, even when you buy it from a company. So for all of my experiments, I typically include a simple control to like calibrate what the transcription rate is on that given day for that stock. And then I use that in my model for simulating the rest of the circuits that were done on that day. So I think approaches like that allow you to sort of say, hey, if this is way slower than I thought, if you have these kinds of controls for looking at specifically transcription rate and translation rate for that day, you can at least reaccount for those differences when you model the system, I guess. Although the models become a lot more complicated there than mine are. Next one, would you be able to speculate how much automation will affect our standards and reproducibility and cell-free? Now we have such reproducibility problems in the field. I wonder if you guys are looking at automation to remove at least some sources of errors. Yeah, so that is, again, ongoing work with Jane. We have like an evolver system that she's going to use to sort of automate the cell lysate preparation process. And then we have a number of small volume liquid handlers, one with positive displacement as well as the echo. And so I think what they want to ultimately do is sort of have some reference plate that you would then test all of your components in for a given lysate and try to use that to understand, where things are going awry. But yeah, that's more of Jane's work. So if you're interested in that, definitely reach out and talk to her. And then next question is maybe a bit far out, but if, when, / hopefully you get to implement this in eukaryotic cells, is there any, yeah, so this is a good one. Is there any concern about double-stranded RNA turning on RNAi pathways? Yeah, I think this is a open question, especially because these things are around the size of what that would be. I typically use double-stranded RNA that's 16 to 22 bases. So it's right around what you would have here. So I think there you'd want to be looking at specifically trying to design sequences that don't have any potential to interact with essential genes or things like that. What would be nice is if there was some way to sort of design the system so that the original component is not the gate itself is not an input, but when you do the reaction, that becomes an input to RNAi pathways, and you could use that to sort of modulate gene expression that way. But I haven't thought too much about exactly how to do that. I'm also not a huge expert on exactly what goes into RNAi, so I just sort of, yeah, I've had that question in the back of my mind, but don't know much about exactly how to modulate it myself. And then I think that was the end of the questions that I saw in the chat.
I think so. I think you answered them all. I was having my own question, but someone already asked it. I was basically curious about the temperature because we saw the same problem when we tried to use ribozymes to control gene expression, just using a ribozyme to cut up mRNA. And it was surprisingly temperature dependent, even within this narrow range that we're allowed to play with in TXTL. So basically between 30 and 37.
Mm-hmm. Yeah, I wonder if some of that is like the folding problem too, that like whatever temperature you're at, you might change, some alternative folding pathway, and then that that messes up the ribozyme. Yeah.
Yeah.
That's why I don't have a great heuristic. I have like just a heuristic of like, if it doesn't work, pick one of these other ribozymes and sort of have a suite of them. But I can't tell you de novo, like, oh, if you have these flanking sequences, this is the best ribozyme to use. I'd like to do that, but it's tricky because those are sort of pseudo-knotted. So a lot of algorithms don't do a very good job of predicting their secondary structure anyway. And then we're folding it while it's being transcribed. So it's like not a thermodynamic problem. There's this sort of kinetic component. So, you know, I think that would be an interesting sort of follow-up study, is trying to do those kinds of, you know, predictive design, but I haven't had the bandwidth to do it.
Yeah. Amaya has a good point. Can that be a feature instead of a bug, building pause to the cascade until you hit track?
Yeah, I think that could be an interesting way of modulating things.
I guess I would work in cells, not in cell free, but I would love to see that.
Yeah, do something like that. Or like, you know, there's a number of papers on building sort of like thermometer type things. And so you might imagine using this, you know, that like if we understood what was going on, that you could do it where certain parts only turn on at a higher temperature, but otherwise are not on, as another way to modulate things. Yeah, that's an interesting point. I hadn't really considered doing anything like that.
If there are no more questions, thank you so much, Sam, again. And thanks, everyone. Have a great rest of your week and see you next week.
All right, thanks, everybody.
Bye.